Hello and welcome to Teach Me in 10. In today's episode, we're diving into the fundamentals of surface plasmon resonance, or SPR, a powerful label-free technology that lets you measure biomolecular binding events in real time. Joining us is Dr. Christin Radon, a Biacore and Reagents application specialist at Cytiva. Together, we'll talk through how SPR works and why it's so valuable in modern drug discovery. Hi, Christine. Thanks so much for joining us to talk about the fundamentals of SPR and why it plays such a critical role in modern life science research. But first, I was wondering if you could tell us a little bit about yourself.
Hi, Laura. Thanks for inviting me. Hi, I'm Christine. I'm Biacore application specialist at Cytiva. I hold a PhD in protein biochemistry and did a lot of structural biology, specifically cryo-electron microscopy during my postdoc. All time across my research, one challenge always came up again and again. Knowing that molecules bind is not enough. Understanding how they bind is what enables confident decisions. I've seen interactions that have identical affinities behave very differently in functional assays. What explained that difference is usually binding kinetics, especially stability and lifetime. If you don't understand kinetics, you're often making decisions with only one part of the picture.
Lovely. And then for someone completely new, what is SPR in simple terms and how does it work?
Okay, so SPR, surface plasmon resonance, lets you watch molecules' interactions as it happens. Instead of only telling you that binding happened, it shows you how fast molecules bind, how fast they come apart, and how long they stay together. All of this is measured in real time and without the need of labels. For researchers, this means you're not guessing from an endpoint, you see the full interaction process as it happens.
Lovely. And could you go a little bit deeper into how SPR actually works, like what's happening at the sensor surface?
Yeah, so in simple terms, there are three parts working together: a sensor chip, microfluidics, and an optics. A molecule is attached to the sensor chip surface, and then another molecule flowing over it in a very controlled way. If binding happens, the molecular mass on the surface is increasing, and that changes how light is reflected from the gold surface. The instrument converts that into a curve called sensogram. And this curve shows binding and unbinding over the time, so you can measure association and dissociation directly.
Amazing. And what kinds of interactions can SPR measure?
So Biacore SPR is incredibly versatile, and it can measure interactions between proteins, antibodies, small molecules without any limitation in size, DNA, and more. It is used either with purified samples, but can be used with serum or complex samples as well. Researchers use Biacore SPR to answer many different questions, from simple yes-no binding to more detailed kinetic analysis, concentration analysis to know the active proportion of your samples, epitope binding, and comparability or biosimilar studies. Biacore SPR is widely used across pharma, biotech, and academia, so with strong support from more than 60,000 peer-reviewed articles. So this broad adoption reflects its role as a trusted tool when detailed interaction insight is needed.
All right. And you mentioned there that Biacore SPR is often used with complex samples. What makes that possible and what practical steps helps avoid issues like high non-specific binding?
Yes, so Biacore SPR detects binding only very close to the surface. Most of the bulk components, which are present in complex samples, like serum proteins or media components, mostly pass through without contributing to the binding signal. And you can reduce issues with a few simple steps, by either clarifying or filter your samples before putting it onto the system. Use reference surfaces and optimize buffers to reduce non-specific binding to the background, and keep protein density on the chip surface under control. Controlled microfluidics also help. They reduce random sticking to the surface and support stable baseline even in dirty samples. Most problems usually trace back to sample preparation or surface design and not to the complexity of the sample itself. So we have a lot of published examples of Biacore being used with patient serums, food samples like milk, and plant-derived extracts for allergen or biomarker work.
Lovely. And could you walk us through what a typical SPR experiment looks like from start to finish?
Yes, of course. So a typical Biacore SPR experiment is very structured and straightforward. And once the workflow is set up, it's actually quite simple to run. So first step is that you prepare your sensor surface. One interaction partner often referred to as ligand, is immobilized or captured on the sensor surface. Modern Biacore systems guide you through this step by step, so you don't need to design everything from scratch. The next step is that you set up reference surfaces and basic controls. These are standard steps and are built into the workflows, helping you to separate real binding from background effects. As a next step, you inject your sample, usually as a concentration series. The system controls the flow automatically, so once a method is set up and defined, the measurement runs hands. As the sample flows over the surface, binding is recorded in real time and showing association and dissociation of the bit complex. If needed, the surface can be regenerated, which allows you to reuse the sensor chip for multiple times and for more than just one measurement. Finally, when you have the data, it can be processed and analyzed. Referencing, curve fitting, and quality checks are integrated into the software, and this helps you quickly see whether the data make any sense and which parameters you can trust. So SPR is a powerful technique, and although it sounds complicated at first, using it in a day-to-day work, it is not complicated at all. With guided workflows and automation, researchers can move quickly from their sample to interpretable decision-ready results.
So it sounds, sort of a simple guided workflow, but what are some considerations someone can have when running SPR experiments?
That's a really, really good question, because this is sometimes what you're stumbling upon when you work, actually. So the theory is, or the principle is, know as much as possible about your molecules. When you work with proteins, know how pure they are, know the oligomeric state, if they're from aggregates, if they're functionally folded. When working with chemical compounds or fragments, know about the purity and the solubility at the working concentration, and maybe as well the mode of action. Is it an inhibitor? What kind of inhibitor is it? So when working with crude extracts, think about good controls to be able to tell real binding and eventual non-specific background binding apart.
Christine, you shared some great insight into how SPR works in practice, but what's your top tip for someone just starting out with SPR?
So what I used to do when I learned Biacore, I was start simple. Begin with a well-characterized interaction. So you can learn how the system behaves and how to read the data with confidence. Always include proper controls. In the beginning, they may feel like extra work, but they will save you a lot of time later on and help you trust your results. Before focusing on kinetics or affinity analysis, spend more time optimizing the surface and the referencing. A clean, well-behaved surface with good controls make everything downstream much easier than trying to fix problematic data during analysis. What is also important is that you know that you don't have to figure this out on your own. Biacore SPR systems include predefined and guided workflows, and your local application specialist can support you in your first steps and later on as well, from service design to data interpretation. Trainings and hand-on support is available to help you build confidence step by step. Starting simple, so to just sum up, starting simple, use available workflows, and get the right support early on is the fastest way to become very, very effective with SPR.
Perfect. And now speaking sort of the broader context of modern drug discovery, why is SPR particularly useful for studying both simple interactions and today's more complex modalities such as bispecific antibodies, ADCs, or macrocyclic peptides?
Yes, okay. So Biacore SPR works for simple interaction because it gives clear qualitative and quantitative information. Does it bind? How stable is the binding? And what are the kinetics? For complex modalities, it becomes even more valuable because it's real-time and label-free. You can observe multi-component binding behavior, avidity effects, and sequential or cooperative binding, things that are often hard to see when you use endpoint or labeled assays. Many complex interactions are usually heterogeneous or don't fit a one-to-one binding model. SPR makes that complexity visible so you can make better decisions.
Perfect, makes sense. And then lastly, to wrap up, what makes SPR well suited for modern research workflows today?
At the moment, modern SPR is designed for today's workflow reality. So many projects, many users, and fast decision needs. One key topic is always the flexibility. So what we just introduced recently is a cup tag capture kit, which enables attaching a cup tag to many different proteins for direct capture on cup surfaces. That is very, very helpful when a facility supports many different targets and projects. It simplifies surface preparation and makes day-to-day work easier. So this matters especially for core facilities, university technology centers, or incubators, where different labs share the same instrument, always different people are on the instrument, and always everyone needs to set up their own experiment. So a standardized workflow helps to keep consistent results consistent across users. With API extensibility, Biacore data can fit into broader digital workflows, supporting integration with data systems and customer analysis pipeline, specifically when companies have different sites all over the world and they share the same database where they analyze the data.
That's great. Christine, thank you so much for taking the time today to talk us through the fascinating science behind SPR.
Thank you for having me.
Thank you. Bye-bye.
Bye-bye.
Thanks for watching this episode of Teach Me in 10 on SPR Fundamentals, brought to you by Cytiva. If you found this video helpful, be sure to like and subscribe to stay up to date with all of our future releases. See you next time.